48%. That's the average share of Native American DNA carried by Mexican Americans today, according to the 1000 Genomes Project. Compare that to 25% for Colombians in the same dataset. Compare it again to just 13% for Puerto Ricans. Three Latin American populations, sequenced using the same methods, the same reference panels, the same standards, and yet the genetic weight of indigenous ancestry swings by nearly 4 times depending on where your family comes from. That gap isn't random. It traces back to one region and one population within it, smaller and quieter than almost anyone would guess. This isn't just a story about modern Mexico's genetic makeup, it's a story about a signal, a specific, traceable strand of ancestry that moved south into the Amazon roughly 6,000 years ago and moved north into California roughly 1,000 years ago, centuries and centuries before anyone had a word for Mexican, or for that matter, Mexico. So how does DNA from one corner of Oaxaca end up rewriting the genetic map of 2 entire continents? This isn't folklore. It isn't nationalism dressed up as science. It's sitting in autosomal DNA panels, in mitochondrial haplogroup frequencies, in Y chromosome lineage studies, published across dozens of peer-reviewed papers by geneticists who, in most cases, weren't even looking for it. They found it anyway, buried in genome after genome, from Alaska to Patagonia. And the number at the start of this, that 48%, isn't where the story begins. It's closer to the end. To understand why Mexican-American genomes carry almost 4 times the indigenous ancestry of Puerto Rican genomes, you have to go back past colonization, past empire, past the rise of agriculture itself. The answer starts almost 13,000 years earlier than anyone expected. If population genetics rabbit holes are your thing, hit subscribe, because this one's got receipts from Oaxaca all the way to the Amazon. Here's what most people assume when they hear the phrase native Mexican DNA. They picture something contained, a single ancestry group, boxed in by the modern borders on a map, distinct from whatever's happening in Peru or Brazil or the Caribbean. That assumption doesn't survive contact with the actual genomic literature. Researchers studying the peopling of the Americas don't treat the continent as a set of separate national genetic pools. They treat it as one continuous, branching system. And within that system, Mexico keeps showing up as a crossroads rather than a container. A whole genome study led by Avila Arcos and colleagues sequenced 76 individuals from 27 indigenous Mexican groups and found average Native American ancestry above 97%, among the least admixed, most genetically continuous populations sequenced anywhere in the hemisphere. At the same time, those same indigenous Mexican groups aren't one uniform population either. They split into deep internal branches. Arido-American populations to the north, Mesoamerican populations to the south and center, a divergence dated to somewhere between 4,000 and 9,900 years ago. Put those two facts side by side and a paradox appears. The more genomes researchers sequence out of Mexico, the less it looks like a genetic endpoint, and the more it looks like a place events radiated outward from. Not a destination, a source. That hub status has a name attached to it, and it's not Aztec and it's not Maya. The group is the mixe, an indigenous population from Oaxaca in southern Mexico. Small today, linguistically isolated, easy to overlook next to the empires that get taught in history class. But ancient genome research, led by S.K. Willerslev's team, detected ancestry closely related to the modern mixe spreading into South America roughly 6,000 years ago. The same team traced a related signal moving in the opposite direction, into North America, roughly 1,000 years ago. Neither of those movements looks like conquest. In both cases, the incoming ancestry mixed into populations that were already established, layering onto existing communities rather than replacing them outright. This is a two-directional genetic signal, originating from one small southern Mexican-related lineage, turning up in the Amazon basin and in the North American interior at 2 completely different points in time. The scale is what makes this strange. This isn't the genetic footprint of an empire. It's a population smaller than most modern cities, and its signature is detectable across thousands of miles and thousands of years. Here's the detail that doesn't fit a simple story. Those 2 migrations aren't part of the same event. They're separated by roughly 5,000 years. Whatever population carried this ancestry didn't just move once and settle. It kept moving, generation after generation, long after the first peopling of the Americas was supposedly finished. To understand how a signal like that survives 13,000 years of continental churn, the story has to go back to Beringia, Ancestral Native Americans diverged from Siberian and East Asian populations around 25,000 years ago, give or take 1,100 years, according to genomic estimates cited in Moreno-Mayar's 2018 study in Science. That's the starting split, the moment the lineage that would become every indigenous population in the Americas separated from its Asian source. A second split followed not long after, somewhere between 22,000 and 18,100 years ago. This time, the divide ran between the population that would go on to become all living Native Americans and a separate branch known as the ancient Beringians. The clearest evidence for the ancient Beringians comes from a single genome, USR1, an infant recovered from Upward Sun River in Alaska, dated to roughly 11,500 years ago. What makes USR1 significant isn't just its age. Its lineage split off before the ancestral Native American population had even fully formed, which means it's a genetic dead end. No living descendants trace back to it. That single fact reshapes the whole picture. There wasn't one clean wave of people entering the Americas. There were multiple, deeply diverged branches, and only some of them left anyone behind at all. The branch that did survive split again, dividing into what researchers now label the Northern Native American and Southern Native American lineages, roughly 17,500 to 14,600 years ago. One of those branches produced the most famous ancient skeleton in North American archaeology. Anzac I is the name given to an infant burial recovered in Montana, dated to roughly 12,900 to 12,700 years ago, and associated with the Clovis toolmaking culture. It's one of the earliest well-preserved ancient genomes recovered anywhere in North America, and for years it served as the direct genetic reference point for the entire Southern Native American branch. Researchers used that genome to test a specific question. Could Clovis-related expansion explain the peopling of South America? The method was straightforward. Check how much later South American ancient genomes actually share ancestry with Anzac I specifically. The finding didn't support a clean answer. The overwhelming majority of later South American ancestry shows no specific affinity to Anzac I at all. That result rules out the idea of one single uniform founding population sweeping down through the continent behind the Clovis toolkit. Instead, the data points towards something more layered. Multiple sublineages appear to have arrived in South America, some carrying Anzac I-like ancestry, some carrying none of it, with the Anzac-related component ending up a minor, minimal long-term contributor to the genetic makeup of the continent. Clovis culture and Clovis genetics, it turns out, don't map neatly onto everyone who came after. That complication needed a bigger data set to resolve, and in 2018, it got one. Two separate research efforts combined 64 of these ancient genomes and turned that single confusing result into a continent-wide picture. Those two 2018 studies, one published in Cell, led by Poth and colleagues, the other in Science, led by Moreno-Mayar and colleagues, pulled together dozens of new ancient genome samples spanning the hemisphere. Both independently found that ancestral populations expanded rapidly across North and South America, starting around 13,000 years ago. But both studies also turned up something less expected. After that initial expansion, there was large-scale movement between North and South America that continued for thousands of years afterward. Not a single founding event, but an ongoing current of people moving back and forth across two continents. That combination, a fast initial spread, followed by continued long-distance movement, is exactly why a single regional genome like Anzac I can't predict the ancestry of everyone who came later. The population was never static long enough for one genetic signature to dominate. Cosimo Post, an archaeogeneticist at the Max Planck Institute for the Science of Human History, noted that the two research teams may have been detecting versions of the same underlying signal, seen from different analytical angles, though a direct comparison of the raw data was still needed to confirm that. This ongoing movement is the backdrop the Oaxacan-linked signal steps into. It isn't part of the first wave. It's a much later current, moving through a hemisphere that had already been settled for thousands of years. That later current is where the Oaxacan connection resurfaces, and where the data gets genuinely strange. Modern genomic modeling of the mixe, used by researchers as a proxy for ancient Mesoamerican ancestry, turned up a signal that didn't fit cleanly into the established framework of ancient Beringians, Northern Native Americans, or Southern Native Americans. Something else was in the data. Researchers modeled that extra signal as gene flow from an unsampled, deeply diverged outgroup. They gave it a working label, Unsampled Population A, or UPOP-A. Based on the modeling, UPOP-A split from other Native American lineages around 24,700 years ago. with a wide uncertainty range running from 30,000 down to 22,000 years ago. The same modeling suggests a pulse of gene flow from this population into mixed ancestors, amounting to roughly 11%, occurring around 8,700 years ago. No genome from this population has ever been recovered. No burial site, no skeletal remains, nothing sequenced directly. Its existence is inferred entirely from statistical patterns detected in the DNA of living people in Oaxaca today. The implication is direct. The native Mexican DNA at the center of this whole story isn't fully explained by any ancient genome science has actually found yet. Part of its ancestry traces back to a population that has never been located. Whatever that ghost population contributed, its descendants didn't stay put. Willerslev's ancient DNA research team detected ancestry closely related to the present-day mix of Oaxaca spreading into South America starting roughly 6,000 years ago. That's thousands of years after the initial expansion into the continent that began around 13,000 years ago. This wasn't part of the original founding wave. It was a distinct, later pulse. The mechanism described in the research is admixture, not replacement. Incoming mix-related ancestry mixed into communities that were already established across South America, layering onto existing populations rather than displacing them. That kind of layered migration is exactly what explains why South American genomes carry multiple distinguishable ancestral components instead of one uniform founder signal running through everyone equally. It also means something specific about timing. Oaxacan-related ancestry became embedded in South American indigenous gene pools millennia before anything resembling Mexico, as a place or an identity, existed anywhere. Researchers studying this signal have flagged that both Willerslev's team and the researchers behind the cell paper may be seeing versions of the same underlying movement, simply described from different analytical angles. Move the clock forward nearly 5,000 years, and the same lineage turns up going the opposite direction. The same broadly mix-related ancestry that moved south 6,000 years ago is also detected moving into North America roughly 1,000 years ago, according to the same research team's findings. Two directions, two very different time frames, one underlying lineage. Separately, a research team led by Nakatsuka sequenced ancient DNA from 79 individuals recovered from archaeological sites across central and southern California. Their results show large-scale movement of genetic lineages characteristic of ancient and modern populations from northwest Mexico into California, dated to roughly 5,200 years ago. An earlier, distinct northward pulse, tied more to language than to the specific mixe signal. That migration connects to the spread of the Uto-Aztecan language family, a language group that today stretches from Shoshone spoken in Idaho, to Nahuatl in central Mexico, to Pipil as far south as Costa Rica. The genetic clustering method used in the study identified a regional movement that hadn't been documented before, prompting researchers to reconsider how and when Uto-Aztecan languages actually spread across that enormous range. Chumash individuals recovered from California's Channel Islands, sampled at roughly 200 years old, still carry shared ancestry, traceable back to that older, northwest Mexican-linked movement, genetic continuity holding steady in one region for thousands of years. Not every indigenous genetic story in the hemisphere runs back to Mexico, though. The Caribbean tells a different one entirely. Ancient DNA recovered from 45 individuals in pre-contact Puerto Rico, published in Molecular Biology and Evolution in 2020, found two mitochondrial haplogroups dominating the sample, A2 at 40% and C1 at 44%. That distribution points somewhere specific. It suggests a primarily Amazonian, South American origin for the ancestors of Caribbean indigenous populations. Not a Mexican origin, not a Mesoamerican one. A completely separate current, running through a completely separate part of the hemisphere. The pattern isn't confined to the ancient sample either. It mirrors the Native American mitochondrial DNA diversity still found in present-day Puerto Rican and Caribbean islander populations today. 3 specific mtDNA haplotypes identified in that pre-contact sample persist in modern Puerto Ricans and other Caribbean islanders right now. Direct maternal line continuity spanning contact, colonization, and 5 centuries afterward. Researchers also found autosomal similarity between the pre-contact Puerto Rico individuals and ancient Bahamian remains, pointing to a shared indigenous Caribbean ancestry component that stands apart from mainland Mesoamerica entirely. The honest picture here matters. This hemisphere's genetic story was never a single Mexican-centered radiation reaching outward in every direction. It's multiple regional currents, Mesoamerican, Amazonian, Beringian, that sometimes overlap and sometimes simply don't. Where Mexico's genetic weight becomes undeniable isn't in the ancient DNA at all. It's in the math of population collapse 500 years later. Romero Hidalgo and colleagues, publishing in Nature Communications in 2021, analyzed 716 newly genotyped individuals drawn from 60 of Mexico's 68 officially recognized indigenous ethnic groups. Their central finding, the genetic structure of Mexico's indigenous populations, is shaped strongly by geography. Populations cluster genetically according to where they actually live, not simply according to shared language family. Two broad groups emerge from that structure: Arido-American populations, the northern, desert-adapted groups, and Mesoamerican populations, centered in the central and southern farming regions. Genetically, these two groups diverged somewhere between 4,000 and 9,900 years ago. That window isn't arbitrary. It lines up almost exactly with the known archaeological timing of when sedentary farming began in Mesoamerica, genetic history and agricultural history converging on the same era. The same study also found something more somber: evidence of population size decline across every single tested indigenous group, spanning the last 15 to 30 generations. That's a demographic signature of colonization-era collapse, still readable directly in the genome today. When researchers compared ancient genomes like ANZIC 128 and USR1 against this modern Mexican indigenous dataset, they found high genetic affinity between them. That affinity reinforces something significant. Mexico's indigenous populations sit unusually close to the deep ancestral source lineages of the entire hemisphere. Then European contact arrived, and the story stopped being about ancient migration and started being about demographic scale. The numbers behind that shift are stark. Pre-Columbian population estimates cited in the 1000 Genomes reconstruction put roughly 14 million people in central Mexico, compared with about 3 million in Colombia, and only around 110,000 in Puerto Rico. Puerto Rico's own pre-contact population figures vary considerably across the historical record. A 1029 Spanish account reported as many as 600,000. Other estimates put the number as low as 30,000 by 1508. Every source, regardless of the exact figure, agrees on one thing: it was a fraction of central Mexico's population. That population gap didn't just shape the history books. It shaped the genetic math of everyone descended from these three regions today. Modeling of post-contact effective population sizes found that the Native American ancestral population behind modern Mexican Americans, the MXL group in genomic studies, numbered somewhere between roughly 62,000 and 64,000 individuals. Set that beside the equivalent ancestral populations behind the other two groups. Roughly 8,700 individuals behind the Columbian, or CLM, ancestral population, meaning the Mexican American ancestral population was about 7 times larger, and roughly 1,900 individuals behind the Puerto Rican, or PUR, ancestral population, meaning the Mexican American figure runs about 32 times larger still. A larger ancestral population carries more surviving genetic diversity forward and a stronger, more persistent signal into modern descendants. It's a direct, measurable echo of pre-Columbian demographic scale, still detectable in genomes sequenced centuries later. That single number, 62,000 against 1,900, is the real explanation behind the 48% Mexican-American figure from the start of this video. Lay the modern numbers out directly. Autosomal Native American ancestry today runs at 48% in Mexican Americans, 25% in Colombians, and 13% in Puerto Ricans, based on 1000 Genomes project sequencing. A separate study of Colombian genomes from Medellin found an even lower figure for that population specifically, an average of just 18.1% Native American ancestry, alongside 74.6% European and 7.3% African ancestry. It reinforces just how much lower the Native American share runs once you step outside Mexico's borders. Inside Mexico, the range stretches far wider than any of those hemispheric averages suggest. Indigenous identified groups average above 97% Native American ancestry. Mestizo populations in northeastern Mexico average 56%, with individual results ranging anywhere from 27.4% up to as high as 81.2%. A national Y chromosome study covering 659 individuals across 10 Mexican mestizo populations found paternal ancestry running 64.9% European, 30.8% Native American, and 4.2% African overall. That paternal Native American share isn't spread evenly across the country. It drops to just 5 to 33% in northern and western Mexico, but but climbs to 37 to 50% in the central and southeastern regions. The shape of that pattern holds across the whole hemisphere. Wherever pre-contact populations were largest and most genetically diverse, modern Native American ancestry runs highest today. That regional split inside Mexico points to the actual biological mechanism keeping this DNA visible at all. It runs through mothers, not fathers. Across Hispanic and Latino populations broadly, researchers consistently find a sex-biased admixture pattern. European ancestry concentrates on the paternal side, traced through the Y chromosome. Native American ancestry concentrates on the maternal side, traced through mitochondrial DNA and the X chromosome. The Columbian genome study showed this directly. Predominantly European contributions running along the male lineage. Native American ancestry disproportionately carried along the female lineage instead. Mexico's own YSNP data shows the identical pattern at a national scale. Native American paternal ancestry sits at just 30.8%, well below the Native American averages found in whole genome studies of the broader Mexican population. The pattern reflects a specific demographic reality from the colonial era. European men and indigenous women forming the majority of founding unions, repeated generation after generation. Because mitochondrial DNA passes only through mothers, Native American maternal lineages persisted at high frequency across the hemisphere, even in populations where paternal ancestry shifted overwhelmingly European. This is precisely why pre-contact Puerto Rico's mitochondrial haplogroups, A2 and C1, still show up in modern islanders today. It's also why Mexico's indigenous mitochondrial diversity remains so exceptionally well preserved, standing in sharp contrast to the far more European-shifted Y chromosome record. Put together, the demographic engine and the maternal line mechanism explain why this signal never actually disappeared. It simply changed which half of the genome it was riding in. Step back for a moment and trace the full chain in one pass. It starts with the ancient Beringians, known through the USR1 genome, splitting off before anyone else. From there, the northern and southern Native American lineages diverged, somewhere between 17,500 and 14,600 years ago. Clovis-associated ANZIC1 represents one branch of that southern lineage, but on its own, it doesn't explain most of the ancestry that shows up later in South America. The picture was always more layered than one genome could carry. Then there's the ghost population, UPOP-A, contributing roughly 11% of mixe ancestry, around 8,700 years ago, itself splitting off from other lineages, some 24,700 years ago, and never once directly sequenced. From there, that Oaxacan-related lineage moved, south into South America roughly 6,000 years ago, north into North America roughly 1,000 years ago, in both directions, mixing into existing populations rather than replacing them. A separate, earlier pulse carried Northwest Mexican-linked, Uto-Aztecan-associated ancestry into California roughly 5,200 years ago. Its own distinct current, running on its own timeline. Meanwhile, the Caribbean followed a different trajectory altogether, rooted in Amazonian South American mitochondrial lineages rather than anything Mesoamerican, and layered on top of every one of these ancient threads, colonial-era demographic collapse. With Mexico's outsized pre-contact population of roughly 14 million people, leaving a proportionally far larger genetic footprint in modern descendants than Colombia's or Puerto Rico's populations ever could. Every one of those threads converges on the same open question researchers still can't close. Start with the Australasian genetic signal detected in Amazonian groups like the Surui, and confirmed in a 10,400-year-old individual from Lagoa Santa, Brazil. It remains unexplained by any current migration model. No one has identified how or when that ancestry entered the Americas, or which population actually carried it. The UPOP-A ghost lineage sitting behind part of the mixe genome shares that same unresolved status. It's inferred mathematically. It has never been confirmed by an actual recovered genome. Researchers studying this material have been explicit about the gap. Comparing the raw data between Willerslev's team's findings and the findings from the Post and Reich research groups was still needed, as of publication, to confirm whether the two teams had detected the same underlying movement, or two entirely separate ones. Even the Arito-American and Mesoamerican divergence date, sitting somewhere between 4,000 and 9,900 years ago, carries a wide uncertainty range. That range reflects how thin the ancient sample coverage still is across most of Mexico. Zoom out further, and the same limitation applies to the whole hemisphere. Only a small fraction of the ancient genetic diversity across the Americas has ever actually been sequenced. Every new ancient genome recovered so far has reshaped the existing model rather than simply confirming it. The evidence suggests that the map this video just walked through is still being redrawn, one recovered genome at a time. Geography positioned Mesoamerica as a narrow, high-diversity corridor sitting directly between North and South America. Any population moving overland between the two continents likely passed through it, or somewhere close to it. Farming's early adoption in the region, dated genetically to that same 4,000 to 9,900 year window as the Arido-American and Mesoamerican split, allowed larger, more stable populations to take root there earlier than in many surrounding regions. Larger, more stable populations produce more genetic diversity and more resilience against the kind of founder effect bottlenecks that erase variation elsewhere. That's part of why indigenous Mexican genomes today rank among the least admixed and most internally diverse anywhere in the hemisphere. That same demographic stability meant more descendants survived forward into the colonial period and beyond, compounding the region's genetic footprint across centuries rather than losing it to a single bottleneck event. Put corridor geography, early agricultural stability, and post-contact population scale together, and Mesoamerica ends up mathematically overrepresented in the ancestry of modern Latin American populations living far outside its own borders today. None of this required conquest. None of it required empire, not in any genetic sense. It's a story of sustained population size and geographic position, compounding quietly over thousands of years. None of that explains everything, though. Some of the oldest signals in this story still don't have a name. The Lagoa Santa Australasian signal sits at roughly 10,400 years old and remains one of the least explained findings in the entire field of New World population genetics. The Upape ghost population behind the mixed genome split off an estimated 22,000 to 30,000 years ago and has left no directly recovered remains anywhere in the archaeological record. Not a bone, not a tooth, nothing sequenced. Even the well-studied ANZIC-1 genome, despite being one of the most complete ancient North American genomes available to researchers, explains only a minority of the ancestry found later in South America. The sample sizes behind these claims remain remarkably small. 15 ancient genomes reconstructing continent-wide migration in one paper, 45 individuals reconstructing the entire genetic history of pre-contact Puerto Rico in another. Every hemisphere-wide model built so far is provisional, standing ready to be overturned by the next batch of ancient DNA recovered from Mexico, from the Amazon, or from the Caribbean. The mixe of Oaxaca today number only in the tens of thousands. Their modern population size gives almost no hint of the outsized genetic role their ancestry appears to have played across two continents, and across 13,000 years. The genome data says the story isn't finished. It just hasn't found the next skeleton yet. A small Oaxacan-related lineage's ancestry surfaces in the Amazon 6,000 years ago, in California 1,000 years ago, and in a 48% autosomal figure among Mexican Americans today. None of that required a single dramatic event. It required geography, farming stability, population scale, and a maternal line mechanism that kept the signal alive through 5 centuries of colonization. The still missing pieces, the Australasian signal, the ghost population behind the mixed genome, the exact overlap between the competing 2018 studies, aren't footnotes tucked at the bottom of a paper. They're the active edge of the research happening right now. If one small population from Oaxaca could leave a genetic mark on 2 continents without ever building an empire, it's worth asking what other population's signal is still sitting unsampled in the ground somewhere, waiting on the next excavation to bring it forward. The field is publishing new ancient genomes on exactly this question on a rolling basis, and each one has, so far, changed the picture rather than settled it. Whatever comes out of the ground next, in Mexico, in the Amazon, or somewhere in the Caribbean, will likely do the same.